So we have an answer already. Okay, no problem. 1 second. Sorry. Yeah. So your answer is introns. Okay. So your answer is absolutely right. Okay. Introns are called as I said, they're called as interrupting sequence, and splicing is a process of removing introns and joining the exons. Correct. So let us go for the next topic, that is genetic codes. So what is a genetic code? Genetic codes are very interesting topics. Why? Because this is where I realized that all the living organisms are connected and all of us are one. So there is nothing called as we are the most advanced or we are the least advanced organisms. All of us have gone, we have evolved from the same ancestry. So this is where all my answers to connectivity and understanding whether there are differences and whether we are special or whether we are, not special, everything was answered from this point of view only. So what are genetic codes? Usually the nucleotides that are present in your DNA are very systematically arranged. Okay, when they are arranged in a sequence, okay, so they actually are synthesized as mRNA, isn't it? So they become a complementary nucleotides and they become mRNA. So mRNA also contains something like, see, you can see if you just read it here from 5 prime to 3 prime, you can just go it like this: A U G U A G A C G U A. When you just read like this, it doesn't make any sense, correct? But when you see in a genetic sense, what happens is every base pair that is present in your mRNA has a meaning. So how does it have a meaning? It forms a code. And these codes are in the unit of 3 codes. Okay, 3 nucleotides combined to construct one genetic code. Okay, and this one genetic code is responsible for assembling a specific type of amino acid for the assembly of proteins. It is such an amazing thing to just even imagine. One small amino acid mistake here and there, your proteins becomes dysfunctional. It either becomes waste, or it becomes hyperfunctional, or it becomes something else. So a very specific organization of this amino acids has to take place, and this instruction is clearly present in your DNA, and the instruction is passed to the cells for the construction of the proteins is in the form of mRNA, and the mRNA contains these codes called as genetic codes. And it was not so easy task to understand the properties of genetic codes or to come to the conclusion of how the genetic codes. Okay, 1 second. This is 1 second. Give me a second, people. Yeah. So when we talk about genetic codes, and the discovery was not so easy. It took a lot of scientific efforts and thinking to come to a conclusion of understanding the properties of genetic codes. That is why mathematics helps us a lot. Okay. So first one is one of the most significant contributions was from George Gamow. So what he suggested was, see, there are 20 amino acids. We know that. So if this 20 amino acids has to be made by your codes, okay, so let us say that there are 3 nucleotides for every code. If there are 3 nucleotides for every code, then there is a possibility of 64 codons. So he will first tell that there are 64 codons coding for 20 amino acids, and it blew up everybody's mind. How can 20 amino acids have 64 codes? Okay, it should have only 20 codes. Now why is it 64 code? Okay, later on what happens? Hargobind Corona. When Hargobind Corona, wait, hold on. Where did it go? Yeah, here it is. There's my point. Students, 1 second. Yeah, sorry. The Hargobind Corona, what he developed, this is very important. Okay, so he developed chemical synthesis of RNA, that is artificial synthesis of mRNA molecule in a definite combination. So that is when the codes were discovered. Okay, this particular sequence of code is giving rise to this particular amino acid. And Hargobind Corona's contribution gave rise to understanding the degenerate nature of genetic codes. What is degenerate nature? As I told, 20 amino acids should have 20 codes, but it has 64 codes. That means more than one code can code for the same amino acid, isn't it? So that happened because of Hargobind Khorona's contribution. Okay, next one is Marshall Neilberg's. So what did Marshall Neilberg do? He's created a cell-free system for protein synthesis. So we understood that amino acids are responsible for the synthesis of protein and how the assembly of amino acids are very essential according to genetic codes. That was done by Marshall Neilberg. And then you have Severo Okaw enzyme, that is, he created something called as polynucleotide, okay, phosphorylase. So what does it do is it polymerizes RNA with definite sequences in a template in an independent matter. You just have to provide, what do you tell? You just have to provide a template using this polynucleotide phosphorylase. The synthesis, a specific amino acid you can just assemble on your own. That means you can synthesize your own mRNA. Okay. So this was the contribution by this scientist. Once we understood that there were 64 codes coding for a genetic code. Now let us look at this genetic code. Okay, there are 20 amino acids, correct? No, so there are certain things that you have to observe when you talk about genetic codes. Okay, look at this one example that I'm going to tell you about. Look at this GUU, GUC, GUA, GUG. What is something that you can observe? You can observe that all the base pairs that are at the corner are different, and all the first two base pairs are same, correct? No. So let us take one more example. GA, 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 GA. Look at here. UCAG. Correct? No? Look at another example. UCAG here. And then we have GGGGG. What is it suggesting? Okay. So what is it suggesting is, look, when you take an example of valine, that is given by 1, 2, 3, 4 different codes. See, 4 different codes coding for same amino acid, isn't it? So these are called as degenerate codes. No, when you observe the characteristics of degenerate code, what you can observe is the first two codes are same. Only the last code is different. Look at glycine. It is the same, isn't it? In glycine case is also the same. Okay, in alanine, sorry, we'll take this out. Okay, we'll take this out because there are 2 different amino acids. You can let us look at this. In alanine case also it is first 2 is same and the last is different. Okay, so we have different amino acids and we have 64 codons. So let us see the characteristics or salient features of genetic code. So the code is triplet. Always the code is in 3 nucleotide sequence, first one. So 61 codes code for amino acids. Okay, and the 3 codes do not code for any amino acids, and these codes are called as, usually I call it as UA, UAG, and UGA. Very easy to remember. Or you can call it as UAA, UAG, and UGA. UA, UAG, and UGA. Correct? No. So these are called as nonsense codons, or they're also called as termination codons or stop codons. And why are they called? Because they do not code for any amino acids and they help in the termination of translation process. So when it comes to the AUG codon, okay, a AUG codon has 2 functions. The first function is it codes for an amino acid called as methionine, and it also acts as an initiator codon. What is an initiator codon? Usually only when there is AUG, okay, only when there is AUG as the first code, okay, then only the translation process can take place. That means the protein synthesis can take place. What is so special here is, whenever the AUG codes for methionine, let us say that, where is AUG? Do we have an AUG here? No, we don't have an AUG. So let us make it as one. We will create our own AUG here. Okay. So we have AUG. Okay. There is one AUG at the beginning. There is one AUG at the middle. So what is the difference between this first AUG and the AUG coming in the between? That you might have thought about it, right? So the first AUG that is actually recruiting methionine is a different type of methionine. We call it as formilated methionine. Okay, formilated methionine. Okay, why formilated methionine or met we'll call it, okay, formilated methionine or methionine. So why do we need formilated methionine? Because it helps in de novo synthesis of amino acid sequences. Okay, this methionine is there, no, the amino acid, if it is formulated, it helps in further assembly. That means it can create the first peptide bonds with the second amino acid. That is why we have formulated methionine in the first AUG. Whereas if the AUG comes in between, is it formulated? Definitely no. It is a normal methionine. That is why we tell AUG as a dual functions, and we have to really understand what is a, see, oh, it's here already, formulated methionine, right? So usually it occurs in prokaryotes, and in eukaryotes it's quite different. Okay, different type of methionine gets assembled there. So that is a different case. Okay, so when it comes to amino acids, correct? No, I told that the codes are degenerate codons. What are degenerate codons? Different codes can code for the same amino acid. So this property is called as degenerate nature of genetic codes. And we talked about how the degenerate nature is there, correct? No. So whenever we write a genetic codes, if you see any of the gene libraries, or if you see any mRNA sequences, that it can be available in the internet. If you just study from 5' to 3', there will be thousands of thousands of nucleotides just typed like this. So all these nucleotides are, once they start from 5' end, they're ending at 3' end. There is no full stop, there is no comma, there is no exclamation or question mark. That means there is no punctuations at all. It is read in a very continuous fashion. Okay, and as I said, the genetic code is universal from bacteria to your protista or any organism you take on this planet. If UUU codes for phenylalanine in bacteria, the same UUU codes for phenylalanine even in humans. Okay, that is the universal nature of our genetic code. Okay, next one is genetic code is non-overlapping. Okay, it doesn't overlap. Next one, it is very specific and unambiguous. Okay, that is one codon specify only one amino acid. Okay, so these are the salient features of genetic code. Okay, people. So we talked about what are mRNA, what is rRNA, and what is we talked about what is polymerase. Let us talk about what is the different types of RNAs that are present before we step into the final process, that is the preparation of your proteins. So mRNA contains a sequence of codes, okay, that are responsible for assembly of amino acids, which in turn helps in protein synthesis. Ribosomal rRNA or ribosomal RNA, it helps in catalytic role, where it helps in recruitment of your ribosome complex, okay, for the process of translation to begin. And the tRNA acts like an adapter molecule. Adapter molecule means it recognizes the specific codes in your RNA sequence. For example, if you have 3 codes in your RNA sequence, your tRNA also has complementary codes or anticodes in it. And then for this code, it will bring a specific amino acid and it will assemble during the protein synthesis. And tRNA has a tremendously important role when it comes to translation. Okay, so we have 5 minutes. Okay. Let's try talking about translation. Should we? Because there is a very less amount of time. Okay, fine. So let's see how much we can talk about it. Okay. So till now, do you have any doubts, students? Do you have any doubts with regard to any topics? We talked about replication and transcription, genetic codes. Are you able to understand the class? Yeah. Yes. Okay. All right. Thank you then. So since we are covering important topics, correct? So that is how the pace of the class will be a little bit faster. But if you have any doubts and if you didn't understand any specific topic, you're always free to put a message in the chat box. So if the time is not sufficient, we can carry on for the next sessions too. Okay. So what is translation, people? Translation is a process of synthesizing a polypeptide chain. In simple terms, it is synthesis of polypeptide chain. So we are not synthesizing proteins, because polypeptide chains and proteins have a lot of difference, because polypeptide chains have to undergo a lot of modifications to become a functional protein. So when it comes to steps in translation, translation is a very elaborate process. Okay, when it comes to prokaryotes or eukaryotes. So what happens in translation is, first one is charging of tRNA. This we also call it as amino acylation of tRNA. That is one process. Second one is initiation, elongation, and termination, as usual, which is same as that same steps as your transcription, but it is not exactly the same mechanism. Okay. So let us see what is the charging of tRNA. First, we have to understand the structure of tRNA before we understand the charging of tRNA. If we observe the structure of tRNA, you can see that the tRNA has 5' end and 3' end. And how do we recognize the 5' end and 3' end of the tRNA? In the 3' end, you have an additional or elongated tail. Okay, there is an elongated sequence of codes. So that end is called as 3' end. And why is the importance of recognizing the 3' end? Because it is also called as amino acid binding site, or it is also called as amino acid acceptor region. Okay, that is first region. So next we have variable loop. Okay, and then we have another loop. See, you can see here. Here we have another loop called as anticodon loop. What is anticodon loop? If I have the sequence like AUG in my RNA, then here what it will be there, instead of A, it will be U, instead of U, it will be A, and instead of G, it will be C in the tRNA. These are two important regions in your tRNA. There are many loops, okay? D loop, okay, Sci-T loop, okay? So we'll study when we are going for the revision class, okay? So I will bring the proper diagram for that. So now, what is amino acylation of tRNA or charging of tRNA? So what happens, an amino acid has to get attached to the tRNA, okay? So that is called as amino acylation of tRNA. So initially what happens is, an amino acid binds to an ATP molecule in the presence of, okay, amino acyl tRNA synthetases, okay? So now this amino acyl tRNA synthetases helps in attachment of amino acid to the ATP. When amino acid has to attach to the ATP, it becomes amino acid and AMP, and it releases two phosphate molecules in the form of pyrophosphate. Before even it attaches to the tRNA, okay, first it has to attach to an ATP molecule, okay? So now when it is attached to the AMP molecule, this amino acyl tRNA synthetase is there? No. This will attach an amino acid and an AMP, okay, an AMP to a specific, okay, to a specific anticode, okay, at the amino acid binding region, forming an ester bond. Now, why do we tell that it is charging of tRNA, or we tell tRNA is charged? We tell this because the same AMP, okay, the same AMP is helping in creation of polypeptide chains. Understood? So it is energizing the reaction of attachment of amino acids. That is why we call it as tRNA is charged. Only when the tRNA is charged, a specific amino acid is assembled to the empty tRNA. You know, you have a truck and you put particular goods and it goes to a specific spot. It's as simple as that. Okay, a specific amino acid is attached to an AMP, and this amino acid and AMP complex is attached to the tRNA. Okay, and then AMP dissociates and it helps the amino acid to attach to your tRNA at the amino acid binding site. Once the tRNA carries this amino acid, we call it as amino acyl tRNA. Okay, we call it as amino acyl tRNA. Understood? So this is called as charging of tRNA. Once the tRNA is charged, okay, now we can go for the further steps of your translation. So first, what happens before the translation has to happen? An initiation should happen. Okay, hold on, sorry. People, I'm sorry, I will just start with initiation, elongation, and termination in the next class. So we have come to the end of the time. Probably it will be late for your next class. So as mandatory, I will have to take an NPS poll. Okay, please try to answer as quickly as possible. Okay. So try to give me a poll as quickly as possible, people. So and the participants, take this as mandatory because it always benefits you in improvising your classes. and it always makes us aware of what's happening with our teaching and all. So that is why it is always necessary to have a review about us. Okay. Excellent. Please feel comfortable talking to me. Okay. I don't feel that I'm that scary. All right. So you can have this comfort and you can always interact with me. And please type your message if you have anything that you feel like asking. Just if you can't ask, put it in the message box. No. So that's what I keep telling. Put it in the chat box. So it's always helpful. Anyways, people, I hope this session was helpful, and I'll see you in the next class, and we'll discuss more about molecular basis of inheritance. And thank you very much for participating in the polls and even in your MCQs and everything else. Okay. So see you in the next class. You can leave the session now.